This is a conversation with Joseph LeDoux. He's a neuroscientist, professor of neuroscience and psychology at New York University, and a rock star, like every other neuroscientist you know. He is the author of multiple books, including The Four Realms of Existence, The Emotional Brain, and The Deep History of Ourselves. He's also the lead singer and songwriter for the Amygdaloids, a rock band that's known for its heavy mental sound. In this conversation, we discuss his journey falling in love with neuroscience and psychology, the split brain experiments. We talk about the amygdala and fear and emotions, and we also talk about free will. This is no time. If you like what you see, then do hit subscribe on YouTube, follow on Spotify, rate 5 stars on Apple Podcasts. This project takes a lot of my time, money, and effort. It's almost like a second job. So if you'd like to see it continue, do consider making a donation on Patreon or Anchor. You can leave a gift on Instagram. You can buy super thanks on YouTube, or you can join and become a member on YouTube. If not through monetary channels, then do consider sharing these episodes, leaving your likes and comments. All forms of engagement, they really go a long way. For other forms of love and support, you can follow this channel on Instagram or Twitter, or follow me personally. And now, it's no time. Like every neuroscientist in the world, you have a double marketing degree. You have a degree in business administration from Louisiana State University with a minor in psychology. And when you were done with that, you thought to yourself, that's not enough. I need to go get a master's in marketing as well. And then along the way, you started falling in love with psychology. And that love came to a forefront when you worked with the legendary Michael Gazzaniga on the infamous split brain experiments during your PhD. I want to start there. What was it about the brain, about neuroscience, about psychology that attracted you so strongly to it back in the 1970s that it pulled you away from the binding hold of marketing?
Well, there wasn't much of a binding hold from marketing. You know, in the late 60s and early 70s, it wasn't very cool to be involved in business. I studied that because my mother said, study that when you go to college. So I said, okay, I just wanted to go to college and have a good time, you know. Yeah. So, I was doing marketing, doing more marketing and blah, blah, blah. And I took a course called Learning and Motivation. I thought that would be relevant to marketing. So turned out it was a course on the brain and memory. The guy was studying rat brains and memory. I had no idea you could do that. So that was a shock. And so I started working in his lab and, I fell in love with the whole process and applied to graduate school. The only place that accepted me was Stony Brook because there was a guy there that knew the guy I was working with and got me in.
Has that love for neuroscience evolved over the last 50 years? You're someone who studied how the brain evolves, how the brain develops. Has your love for neuroscience also evolved? Or do you think the reasons that pulled you in the first place 50 years ago are also the reasons you're in love with it now?
Well, yeah, it's like at some point, it becomes your job, right? Yeah. But I want to go back to one thing, which is you asked about psychology. And it was really psychology that, that pulled me into the whole thing. And the way I think what really helped is that I didn't have training in psychology. I just had some knowledge. And when I was applying to graduate school, I had to take not only the graduate record exam, GRE, but also the psychology advanced test. And I literally read a bunch of psychology textbooks and knew everything. It was all fresh because I was learning it for the test. And I think that really, was a success for me that I was able to do that and get in. I think that helped me get into graduate school.
Any book that you remember in particular?
Yeah, it was a book called Silverman. And he was at this place that I didn't know too much about at the time. It's called New York University, which is where I ended up working. So, and when I got there, I asked people, did you know this guy Silverman? There were still some senior people around at that time that were in the psychology department. And they said, yeah, we knew him. But he was long gone. Nobody there would know who that is. That's a big problem with science today is young people don't know more than like 2 or 3 years back. It's like always about the next technique now. It's not about what are we going to discover. It's about how to, what's the coolest technique I can do.
There are a lot of trends that come into place. Okay, we've already established that like most neuroscientists, you have a double marketing degree. Just like every other neuroscientist in the world, you also moonlight as a rock star. You're the lead singer and songwriter for the rock band, the Amygdaloids. I think you're one of the only people in the world who can answer this question truthfully.
Okay.
Does does studying the brain, does working like a neuroscientist give you any hacks into becoming a better musician?
Well, it allowed, I was a musician in high school as a kid, and I was always very attracted to pop music. So it was something I really cared about. But I wasn't good enough to actually, we had a band, but, there was no future for me as a musician, really. And it is only neuroscience that made it possible for me to have the band, because we created this genre called heavy mental, music about mind and brain and mental disorders. And that was kind of, catchy. And we got a lot of invitations. We played at Madison Square Garden for NYU's graduation, all kinds of stuff. The Kennedy Center, written about in the New York Times, all kinds of magazines. This all started in 2006. And, we still occasionally play from time to time. We released 5 or 6, maybe 7 albums that I remember. One of them, I had Roseanne Cash as my backup singer. And so we've had a great run.
So working as a neuroscientist did influence that genre of music that you created. But do you think studying fear, knowing what happens on a neural level when you dissect fear, do you think that has helped you combat fear in your life, like combat stage fright?
I think as you get older, I guess it can go 2 ways. You can become more fearful or less. And I feel like I'm less fearful these days. Maybe a little more anxious. I don't know. There's a difference.
I guess I don't know if it net worked out or not. Does it work the other way? Does working as a musician, creating music, writing songs, does that give you a fresh perspective that helps you in your research?
Not really.
No transferable skills at all.
It doesn't go in both directions. It would be nice if it did, but truthfully, it's, I just use things I knew about the brain, mind, philosophy to come up with songs. Like one of our songs is called Theory of My Mind. Yeah. So that's based on theory of mind, obviously. But it's, all these songs are love songs about mind and brain and mental disorders. Yeah. And like most rock songs are about mental disorders in a way, or at least mind and brain. So yeah, the Theory of My Mind was about a romance. And do you have a theory of my mind? If so, why are you so blind to all the things I do and blah, 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 that kind of stuff.
So if there's any aspiring neuroscientist out there who also wants to be a rock star, you're saying no transferable skills.
Well, we might become a better musician.
A small glimmer of hope. Okay. Unfortunately, the fun questions are out of the way. Let's dive into some of the heavy lifting. Let's talk about some of your research. I think the best way to set the context for everything we're going to talk about is by summarizing your latest book, The 4 Realms of Existence. So let's create the big picture view, and then hopefully we can start narrowing down to specific areas of your research. So first, let's talk about this. What are the 4 realms of existence? What is the new theory of being human?
Well, it's not some Eastern mystical thing, as a lot of people expected it to be. It's a pretty boring set of 4 things. The basic idea is that all the organisms that have ever lived are biological organisms. That goes, if it's alive, it's biological. If it's not alive, it's not biological. Yeah. So some biological organisms developed a nervous system, and that happens to only happen in mammals. in animals. Yeah. So only animals have a nervous system. All other organisms alive are biological, but not neurobiological. So neurobiological is the second realm. Yep. And then the third realm is that some of these organisms with nervous systems have cognition. They're able to think and plan and make decisions on the basis of information in the head rather than information necessarily just in the world. Yeah. So that, it's very easy to say which organisms which things in the world are biological. If it's alive, it's biological. It's very easy to say which organisms are neurobiological. If it has a nervous system, it's neurobiological. But when it comes to the next 2 realms, the cognitive being the next one, then we have a problem, because there's no functional structural marker that tells you this is a cognitive creature. Yeah. You have to do rigorous tests behavioral tests to try and pin it down. Mm-hmm. So the rigorous tests that have been done have, I think, pretty conclusively told us that mammals and birds are cognitive creatures. It's hard to say in other vertebrates, like reptiles. It's a little more difficult to like say whether they are or not. I mean, it doesn't seem that they have the same thing that mammals and birds have. And when you get into invertebrates, again, it doesn't seem they have exactly the same thing. Maybe they have their own version of it. But if we're looking at this from the evolutionary point of view, it basically started with mammals as one path, and in birds through another path, through reptiles that the other reptiles didn't develop. And not even all birds have developed this. Only some birds, like shrub jays, it's like corvids, they're called. Yeah. And so these capacities happened in parallel, not in sequence. There's not an ancestor that gave rise to cognition in birds and mammals. Okay. So that we have to develop tests, and there have been some very good tests that tell you a behavior that is not cognitive is a habit. So a habit is something that you've learned. It's become routinized so much that in the presence of the stimulus, the behavior is performed. There's no intermediating cognition, thought, plan, action, decision in the performance of that habit. It's just a stimulus-response connection. So the distinction that we want to make here is between habits and goal-directed behaviors, behaviors in which you have to plan and make decisions to achieve the goal. The goal doesn't just come to you as a reinforcer habitually. You have to make it happen by having a plan that can conclude an action that is useful to you. So that's the main difference that we can draw, that the thing that birds and mammals can do and other creatures are not as good as, or maybe not even capable of. Mm-hmm. So that takes us to the fourth realm, which is the conscious realm. And again, we have no physical marker that tells us, what consciousness is and which animals have it. And it's even more difficult to know which animals, even amongst mammals and birds, or even if we take those two as like the canonical examples, we don't know if any other mammal has consciousness or if any birds have it. The way I like to think about it is that consciousness depends on cognition, and the cognition takes you to the finishing line. And that means that we can study things like, episodic memory and semantic memory in other animals. We call it episodic-like rather than actual episodic, because we don't know that the animal has the internal representation and the conscious experience. You can use cognition as a guide, because what it does is it takes us all the way to the finishing line where consciousness can occur. But we don't know if other animals cross the finishing line. But it's pretty good to even be able to say that these are markers that we can see how they get to the finishing line. And, this is a way to think about, do these other creatures like, bees and flies and so forth, do they even get to the finishing line? Or are they, somewhere else? Yeah. So that's why I like that particular distinction. So in terms of human consciousness, I mean, there's so much debate in the field and so much we could talk about. But I'll tell you my particular position on that, if you like.
I would love to hear it.
So, well, I guess we should go back to something you mentioned earlier, which is the work I did for my graduate thesis on split brain patients. Now, split brain patients are people in whom the two halves of the two hemispheres are sectioned, the connections between them are sectioned, to relieve intractable epilepsy. This is not something that you do lightly, obviously, because you're disconnecting one hemisphere from the other. But it seemed, in the 1970s when we were doing these studies, I mean, we weren't doing these surgeries, obviously. There was a surgeon at Dartmouth that was doing it, and there was a surgeon in Southern California that had done it in the 60s. And my mentor, Mike Zanica, worked on those patients in the 60s when he was at Caltech as a graduate student. So it was only 10 or so years after that I joined him. So he wasn't that much older than me. So we got along quite well. So we were studying these patients not to understand their epilepsy, but to understand what goes on in the mind when the brain is split. Now, a lot of that Mike had done, he worked out the details of the basics of what happens when the brain is split. So if you show a picture to the left side of visual space, it goes to the right hemisphere. If you show a picture on the right side, it goes to the left hemisphere. So when you put information, for example, if I flashed information into your left visual field, it would go to your right hemisphere, and I would say, what did you see? You would say, oh, I saw an apple. Okay, if that's what I showed you. A split brain patient, when you put information on the left side of space, it goes to the right visual field. I'm sorry, it goes to the right hemisphere, but the left hemisphere can't talk about it. Left hemisphere is where you speak, right? Yeah. But it has no access to what that right hemisphere is holding, what information it has. But the right hemisphere can, using the left hand, pick up the apple and say, this is it. I mean, he doesn't say it, he shows it, points to it, you know? Yeah. And then the left hemisphere can say, oh, yeah, I knew what that was, and kind of make up a story. And that was a key thing in our experiments. The way that these patients made up stories to compensate for the fact that the right hemisphere has information that the left hemisphere doesn't. And so the left hemisphere, we know, has a active conscious mind because it's talking to you and keeping up with the conversation, knows what happened in the past, has predictions of the future and all of that. But we don't know about the right hemisphere, what's going on over there. And so we were very curious about these kind of confabulations that the left hemisphere would generate in the faced with having just produced a behavior. So we designed some experiments to kind of test this. I mean, in this one patient that we were studying, his initials were PS, patients always get a name like that. His name was PS. And we had the opportunity for the first time to ask an important question, which is the right hemisphere conscious? When you can only use pointing to things as, as the indicator. Yeah. You can't really say much because you don't know if there's like a smart chimpanzee over there or if there's really a human being. Yeah. But we had this one patient, PS, who could both read and talk in the left hemisphere, as all the patients could. But in addition, he could read in the right hemisphere. That was very unusual. He couldn't talk out of the right hemisphere, but he could read. So this, for the first time, we could probe whether there was a person over there by asking it verbal questions. So we put in, who are you, in the left visual field. He goes to the right hemisphere, and he spells Paul with his left hand. So it's going over there, and it's able to answer by spelling it out with Scrabble letters. We just list, it'd be like Scrabble letters. He pulls them out, P-A-U-L. And that was kind of chilling. It was like spine tingling because here was a part of the brain that normally could not communicate with you in any kind of human sense. But he knew who he was. He knew his name, Paul. He also had he had social relationships. If you asked him who his favorite person was, he said the Fonz from Happy Days. If you asked him who his girlfriend was, he spelled out Liz, L-I-Z, his girlfriend's name, and on like that. So he had a sense of self. He had a social sense. And when we asked him what he wanted to be when he grew up, the right hemisphere said he wanted to be an auto racer. But the left hemisphere in conversation would always tell us he wanted to be a draftsman. So two different, same person really in many ways, but two different views of the future. I mean, that's fascinating, right? Now, how much, weight can we put on this? So we got one patient, and he's doing some really cool stuff, telling us some really important things. But, it's hard to know whether, how far to take this. But it was so clear in him. It's not like you need it's not like you needed a statistical test to see these kinds of things. These were not like, a group of patients. It was like, you could see that this is really what was going on. Obviously, the right hemisphere could read. Yep. And because he could read, he could generate these very complicated, important kinds of indications that he was conscious. Yeah. So the kind of the coup de grace test that we did, we presented two pictures simultaneously, one to the left and one to the right. In the left visual field going to the right hemisphere, was a snow scene, snow, car covered in snow and stuff. The right visual field going to the left hemisphere was a chicken, or chicken claw. Sorry, chicken claw. And so then the left hand goes to a shovel, and the right hand goes to a chicken. So we said, why did you do that? Now we're talking to the right hemisphere. I'm sorry, we're talking to the left hemisphere. So we said, why did you do that? And we're talking to the left hemisphere at this point. Yep. And so he says, well, I saw a chicken claw, so I pointed to the chicken, and you need a shovel to clean out the chicken shed. So he observed the behavior of his left hand and came up with a story to make his left hand and right hand choices fit together in a seamless narration. There wasn't a split second of thought. It was simply when we said, why did you do that? He said, well, I saw the chicken claw, so I pointed to the chicken and need a shovel to clean out the chicken shed. So the instant thing, at first we thought, well, this is, again, confabulation. But, and neurological patients often confabulate to take care of their problems or to compensate for their problems. But, so what? What we concluded, we'd go to the bar every night in Vermont after we were testing the patients and drink a little Jack Daniels and kind of speculate about what was going on. At first we were thinking about, confabulation. Then the light bulb goes off. This is not just something we see in patients because of their brain damage and brain surgery. It's something we do every day, all the time. Mm-hmm. We make up stories to make sense of who we are, of what we're doing. And the basic idea was so Mike was a good friend with Leon Festinger, who had come up with the idea of cognitive dissonance in the 1950s, late 50s. And so, he borrowed from these social psychologists like Festinger very freely, of course, crediting them. But, not many neuroscience researchers at the time were thinking about social psychology, but Mike was always ahead of the curve. So he said, maybe what's going on here is that, we have all these systems in our brain that are capable of producing behavior unconsciously. And it's, because we all believe we have free will, regardless of what scientists think, most people, including me, think we have free will of some kind. So if we're going to protect that sense of free will, we have to somehow get past the fact that we aren't always controlling what we do. So if you are behaving in a way that is inconsistent with what your conscious mind thinks is going on, it makes sense that there's got to be some kind of dissonance reducing narration that makes it all make sense. Yeah. And so that's the idea that we came up over these cocktails after the experiments, and we wrote a book together on the basis of my PhD work and the work that some other students at the time were doing. It was called The Integrated Mind, and that was my first book. It wasn't a blockbuster, but it's it's still around, and in it we had the whole story that I just told you. Yeah. But there was one other aspect of that. The other thing we talked about at the bar one night was that Mike said, it's interesting. We'd done this experiment on that had some kind of emotional component to it in this patient. And so that night after the experiment, we were talking and said, there's not much work on emotion going on in neuroscience right now. You should consider that. I said, yeah, I'm going to do that. The light bulb went off and I decided that's what I wanted to do, because that was another professor in graduate school had told several students that were in his seminar, said, well, when you leave graduate school and go out to find your career, do not study the hippocampus, because there's too many people doing that. And do not study the visual system, because there's way too many people doing that, because those were the big hot topics at the time. So that was on my mind. And so when Mike said, there's not much work in neuroscience on emotions, I'm I'm going for that. So the next step was, how to do that. And there was not really very good techniques at the time for studying, the human brain and psychological functions. Most of the research involved patients with brain damage of some kind or brain surgery. And you had to kind of wait until you had patients of the right type to ask the question or to answer the question that you wanted to ask. And it could take years to gather the data, in that situation. And I was kind of a eager, energetic young man. I didn't have years to wait. I wanted to do it immediately. So I thought, well, there's a lot of thought at this point in time, the early 70s, about emotion systems in the brain. That there'd been some talk in earlier years, but again, there was not a lot of research going on. But there were ideas from the 50s and 40s and so forth. The so-called limbic system theory was very popular and still was at that time. And so the idea was that the human brain evolved these emotional circuits from other mammals, our mammalian ancestors. And so if that was the case, then that would mean that I could study emotional behavior in a rat, and it would be relevant to emotional behavior in a human. I knew there was no confusion in my mind at all. I knew I was not going to be able to study consciousness in rodents, but I did know that I could study the animal's behavior. So I decided to take that task on to go back to what I had done for a few months when I was studying marketing and had done that research that was on rat brains. I don't think I said that earlier. So I went back to rats and started trying to figure out the systems in the brain that would allow the flow of information from a stimulus to go through the brain all the way to the motor system that controls the response. So all the way from the sensory system through the brain to the motor system. Because when you work on split brain patients, you become acutely aware of the flow of information. If you disrupt that flow of information, then everything breaks down. So that was my kind of mental model of how to approach it. Also at the time, researchers like Eric Kandel were doing great things in aplesia, invertebrates, where you had a very simple nervous system and you could trace it from the sensory system all the way through. And it was thought at that time you could not do that in the mammalian brain. It was just too complicated. In fact, Kandel started on the mammalian brain, but says too complicated. And he went to the aplesia, and it took us kind of stuck. It got stuck for a number of years like that. But by the 70s, I was getting started doing this. There were a couple of other labs getting started doing this, different kinds of preparations but it was all using Pavlovian conditioning. And with Pavlovian conditioning, I felt certain that I could follow the stimulus all the way to the response because it was a very simple stimulus. It was a tone paired with an aversive stimulus. And then the animal, would hear the tone, it would then freeze and its blood pressure and heart rate would go up. So, okay. It seemed pretty simple. The responses were kind of innate, hardwired responses. That means there would be a circuit that was controlling those. I just had to figure out how a tone comes into the ear and gets to the circuit that controls the behavior. Simple. And it turned out simple. It was actually pretty simple because at the time, there were new techniques for tracing circuits in the brain. In the old days, you had to make a lesion in the brain, and then the neurons would degenerate, and you'd have to find the degenerating axons to be able to figure it all out. But with the new techniques, you could inject a tracer in a specific brain area. It would be picked up by the neurons and transported down the axon. And you were able to just march through the brain, making lesions of a brain area, let's say in the sensory system. So I started by lesioning the auditory cortex. The animals could still be conditioned when the auditory cortex was lesioned. All right, so what's going on? We lesioned the auditory thalamus. They could not be conditioned. So at that time, the only connection from the auditory thalamus anywhere was to the auditory cortex. That was what was known. But we said there must be some other connection. And the tracer that we injected into the thalamus like that went to several areas in the subcortical forebrain, the hypothalamus, the caudate putamen, the intralaminar thalamus, and the amygdala. Now, the amygdala had been kicking around in the field for a long time, but never as, there was no understanding of how information would go in or come out of it. But we found this direct pathway from the auditory thalamus, the medial geniculum, into the amygdala. And when we lesioned the amygdala, it disrupted conditioning. And at first we started with big lesions of the amygdala, but then we said, okay, the tracer is not going to the whole amygdala, it's only going to one part. So we lesioned the part that was receiving the input, and that disrupted everything. So then we injected the tracer to see what that area of the amygdala that receives the input, where it's going to, and it was connecting to other parts of the amygdala. So we lesioned those, and one of them, the central nucleus of the amygdala, disrupted the outputs. And the connections of that to the motor systems was direct. From there, you went to the periaqueductal gray and then down to the somatic motor system. And from the central amygdala to the hypothalamus and brainstem to control blood pressure and heart rate. So it really only took, 5 or 6 years to do all that. Maybe 7 or 8. I mean, it was relatively speaking, to get from the ear to the muscle is a pretty quick task.
Let me try to simplify this with the thought experiment.
Yeah, okay.
Let's say I'm walking down the street and I see something I'm really afraid of. Let's say a crazy lion, for example. What happens, and you've already covered a lot of this, but in terms of this example, what's going to happen in my body when I receive that visual stimulus to my reaction of freezing up and then most likely running away from my life? On a neural level, what's happening in my body the moment I perceive a threat, and how does fear given rise to in the amygdala?
First thing we have to note is that for the auditory stimulus to go from the thalamus to the amygdala means that it's taking a shortcut, because the stimulus also gets from the auditory cortex to the amygdala when everything is intact. Mm-hmm. So the idea that I came up with very early on was a low road that's imprecise but fast, and a high road that would allow cortical processing to be more elaborate and control the amygdala as well. So when you see a snake or something on the ground that's all of a sudden triggering you, that's going to take the stimulus through the low road to the amygdala and allow you to respond immediately. At the same time, it will more slowly go to the auditory cortex and make its way to the amygdala. But in addition, from the auditory cortex, it also goes to the prefrontal cortex. And the prefrontal cortex, I think, is the answer to how the fear itself comes about, as opposed to the behavioral and physiological responses.
Where would you place fear in these 4 realms of existence? Is fear something that's related to consciousness? Does it come with cognition? Or do you think it goes all the way back to even bacteria, multicellular organisms? Because there's some form of survival instinct, awareness of danger, even in those multicellular organisms. But can they feel fear?
So let's start with, again, in the human brain and work backwards. So
sure.
In the human brain, the stimulus will get to the amygdala in some way. And when it's there, it will activate the behavioral and physiological responses. That circuit I call a defensive survival circuit, not a fear circuit, because fear is something different. Fear is the conscious experience of being in harm's way. And that, I think, is a cognitive situation. In other words, if we go back to the split brain patient who is making up a story to explain his behavior, it's the same thing when we encounter danger in our lives. Stimulus goes to the prefrontal cortex. A story is concocted about that. All emotions for me are concoctions, interpretations. There are no basic and non-basic emotions, or inherited, and there's no inherited emotions. They're inherited responses, but the emotion is a cognitively constructed interpretation of the situation. So now we get back to the question of which animals have that. Or you asked about the 4 realms. So that is what we talked about there is the cognitive and conscious realm generating the experience of fear, and the neurobiological realm generating the behavioral and physiological responses. So these defensive survival circuits are present in all mammals, all vertebrates, and they're also present in invertebrates because they have to detect danger and respond to danger as well. So it's not just something that is in mammals and related to cognition. The defensive survival circuitry is there to detect and respond to danger. So without the cognitive realm, and again, we don't know which, whether bees and bugs and so forth have the cognitive realm. If they do, then they could construct some kind of semblance of what we're talking about here. So, but the ability to detect danger is not simply something that is present in animals. It's not just a neurobiological realm response. We can go to protozoa, the ancestors, the single cell ancestors of animals. Now, protozoa have to detect danger and respond to danger as well. And even though they are one cell, they swim around in the water or wherever they are living, and they detect danger, they turn away from it. If they encounter nutrients, they keep going. They can even learn. They learn to avoid harmful things. Single cell. So learning and behavior have nothing to do with what we call psychology. They have nothing to do with what we call cognition and consciousness. Learning and behavior are ancient. They are present in single cells, including all the way back to the earliest animals, all the way back to the earliest organisms, bacteria. So you have LeDoux here, the first bacterial cell that was able to live long enough to survive and then divide and give rise to the rest of all of life. LeDoux was able to detect and respond to danger and to approach nutrients and stay alive long enough to be able to divide and reproduce, replicate.
I'm going to commit the cardinal sin of asking you to summarize a book here, but you have written a book, A Deep History of Ourselves, How We Got Our Conscious Brains.
The 4 billion year story.
The 4 billion year story. And I'm going to ask you to summarize that 4 billion year story in a 2 to 3 minute answer. So maybe this is for 10 points.
Okay.
But how did we go from LeDoux, the last universal common ancestor, all the way to the human brain that we know
Let me tell you how I got to LeDoux.
Yeah.
So I was, at some point in my laboratory research, I got involved in, we'd figured out the circuitry. So the invertebrate model is once you learn the circuitry, you can then go after the molecular mechanisms. So we knew about the amygdala and the inputs and outputs and all that. So we started studying the molecular mechanisms. And so how do you do that? Well, you look for other research that has studied molecular mechanisms. The work in invertebrates, especially Candel's work, had discovered all kinds of kinases, protein kinases that were important in learning and memory in the aplysia. This information was then used by researchers studying the hippocampus to understand long-term potentiation, a kind of model of synaptic plasticity. And we were using the same information to try to understand molecular mechanisms of so-called fear conditioning. So that meant that something like protein kinase A and MAP kinase and things like that Kendall had implicated in learning in those invertebrates. And the same was true in mammals. So how did that happen evolutionarily? Most likely that means there was a common ancestor 630 million years ago. We know that there was a common ancestor, but the idea is that the invertebrates and vertebrates inherited the same molecular mechanisms, the same genes, and allowed us to borrow from the aplysia. Okay, so I said, okay, we're 630 million years. That's pretty old. That's a long time ago. But where did that come from? So I was digging into that and discovered that this ancestor was all of the animals we've been talking about, vertebrates and invertebrates, are called bilateral animals. They have a left and a right, a front and a back, and a top and a bottom. Before them, before this common ancestor, which had this bilateral organization, very primitive, little brain in the front and like some neurons going to the body and stuff, but it was just basically a flatworm. And before that, we have something like jellyfish, and so there you have a diffuse nervous system. The flatworm has a nerve cord coming out of the brain going to the rest of the body. The jellyfish and others in that phylum have a relatively diffuse nervous system, no centralization. So something happened between jellyfish and this flatworm, and it all has to do with larvae, because jellyfish go through a larval phase where they're not just a big ball and all the tentacles and all of that stuff. It's just like a little kind of worm-like thing, a little larva that is swimming around. And if you look at the ancestor of the animal, it's going to be the larva, not the jellyfish. It's going to be the larval phase, because evolution and development work together. And through the early development of the body, you're dealing with the primitive baseline organism. And then as the body matures and becomes the adult, you then take on qualities that are not represented in the early larva. But it's that early larva that's connected to the genome and then becomes the basis for the extension of new animals, the arrival, the arrival of new species out of that.
So you brought the connection with the larva. Then I have to ask you a popular junk food question. In your book, Synaptic Self, there's a chapter titled Building the Brain, where you say genes dictate that we will all have a human kind of brain with roughly the same kinds of circuits, but random individual differences will exist, and the connectivity of the circuits selected by synaptic activity will shape the individual brain. You just spoke about the larva and the similarity there, and then how they inherit certain features, but then they grow on to develop their own. I have to ask you the age-old nature versus nurture question. Let's bring it back to humans. How much of our brain is genetic, determined by nature, and how much of it is shaped by synaptic activity, shaped by experiences, nurtured during our lifetime?
I think no one has any idea, but it's a good question, but I don't think we know. The way I like to think of the brain is that every cell is basically a bell curve. And so it is affected by every cell that it interacts with, right? And so you have a bell curve for each cell, and then a bell curve for each interaction, and on. You go from that all the way through the entire brain. So all of that is going to start as some kind of genetic programming, but as soon as that starts, you're going to have all these modifications, epigenetic and other kinds of environmental influences that, together with the genes, shape the bell curves all the way through development, and they're still being shaped as we go through life.
So it's a combination of both, I assume, and I hope to get that answer soon. Let me ask you another popular junk food question while we are quickly swatting aside these junk food questions. Baruch Spinoza had once said that in the mind, there is no absolute of free will. You spoke about free will earlier, and you mentioned that you would like to believe in it. Spinoza had said that the mind is determined for willing this or that by a cause, which is determined in its turn by another cause, and this one again by another, and so on to infinity. René Descartes was a strong advocate for what is now known as dualism, this theory that's now dubbed as the Cartesian theater, the little man, the homunculus in your pineal gland, which is your consciousness that is making all the decisions, gives you autonomy. You just mentioned you like to believe in free will. Why do you think that is? Even though most of your research has shown that a lot of our actions, reactions, everything we spoke about now is the result of something that's happening in the brain that's not in a control. We're just generating a narrative around it. Then why do you still want to believe in free will?
So let me give you a very easy answer. Yeah. We are biological organisms that have neurobiological circuitry that underlie cognition. And the next step is consciousness. So determinism can work pretty well for those lower levels, but I'm allowing consciousness, and maybe cognition as well, cognition and consciousness, to possibly have some choices. you mentioned dualism, and
The
there's
mind-body problem.
mind-body problem. There's something called qualia that is supposed to be what it's like to be conscious,
What is it like to be a bat?
and what it's like to be a bat. And so the Nagel Chalmers
Heart problem.
has been that idea has been pushed into neuroscience. So is it hard in the sense that the mechanisms of inheritance were hard before we understood DNA? Or is it hard in the sense of a soul that survives death? One is a scientific problem, the other is a philosophical problem. And I think that is the crux of the difficulty we have when we take philosophical terms, turn them into scientific terms, and then search for the answer to those terms, especially when the originator of the term or the issue is saying that it's a non-physical problem. Therefore, it can't be in the brain. So that's my problem with the whole qualia thing.
Let me ask you one final big question as we approach the end of the hour. So we were talking about fear and the amygdala. Your colleague, Yuri Bushaki at NYU, he was on the show last year, and he had said something interesting where he had said that the boundaries of the brain as we know them are mostly arbitrary mapping. Some are logical, some are random. It's an outside-in approach where we try to find a home for memory, so that must be the hippocampus. We need to find a home for emotions, so that's the amygdala. But rather than the regions, we need to focus on the interactions. The brain works as a whole with no demarcated regions responsible for functions. Would you agree or disagree with Professor Bushaki? Because do you think there's merit to the argument there are specific regions and boundaries that perform specialized functions? Or do you think that's a wrong way of thinking about it? It's wrong to think of the hippocampus for memories, the amygdala for emotions. We should just view the brain as a whole and the interactions between the different regions.
So one of the reasons why I wrote the Four Realms of Existence was because I felt that we had a lot of baggage in psychology and neuroscience. For example, the idea of the self. We have this idea of a self inside us that is doing things, that is controlling our behavior and so forth. But there's nothing besides me inside of me. And I think that we have to, when we reify these concepts, old ancient concepts, we get into trouble. And I agree with Yuri that ultimately certainly there's no emotion in the amygdala. I would put it as a cognitive construction in prefrontal cortex. But all of a sudden, I'm blending cognition and emotion. So I think both of those are made up terms. Those are words we use as humans to understand what goes on in our mind. I'm a big believer in folk psychology as being important. We'll never get rid of folk psychology. I mean, you can scientificize folk psychology all you want, but that just means we'll get other folk psychology to work around that. I think that that we really have to be more aware of the concepts we use and the ways we talk about things, and try to strip them down. And every few years, let's ask ourselves as scientists, do we have it right? Are we really talking about, say, fear when we study fear? Or is that something that we're just kidding ourselves about?
I could keep asking you questions all day, but we are approaching the end of the hour and you'll be mindful of your time. So let's start moving into some of our closing questions. Before we move there, I would love it if you can interpret what I've built with the Lego. What do you think this is? Whoops. It's a masterpiece. I wouldn't.
What am I supposed? I'm supposed to guess what.
What do you think it looks like? Yeah,
it looks like out there.
Yeah, why not? Yeah, this is what I was replicating, the skyline. Well, I don't know if I was building it or there's some reaction in my brain and I was just responding to it. Okay, fantastic. Let's move into some of our final questions. In your book, you say Plato, for example, believed that the intellectual essence of an individual, his psyche or soul, survives death. In fact, Plato looked forward to death so that he could be free of his body and all its needs and passions and finally be capable of pure thought. You're someone who studied the brain extensively, so I think you're the perfect person to ask this question. What happens when we die? Is it possible that Plato was right, that the brain, the consciousness, psyche or soul can be preserved and we can just be capable of pure thought?
Well, I think, I was raised as a Catholic, and I learned, I should say, I was indoctrinated with a lot of stuff by the nuns, but I've spent my life trying to, remove myself. So yeah, I don't believe in life after death. By definition, there's no life after death biologically.
The work that Neuralink is doing, for example, of these brain-computer interfaces, there's this, I guess, this junk food myth being being promoted that you can upload your memories and then download in a future vessel. Do you think that'd be possible with like Neuralink?
Well, maybe some minor version of it, but I'm not a big fan of like consciousness and AI. I think that consciousness is a series of billions and billions of years of biological accidents, and that unless you can replicate all of those steps, we'll never find out exactly what it is. It's like some researchers want to map every neuron in the cortex and assume that they will then understand the cortex. But the neuron is like a very, very blunt thing. It's what's important is all the stuff that's going on in the neuron. And there are, millions of years of history that are driving what those cells are doing deep down molecularly.
Okay, so we'll have to replicate evolution and those biological properties so many billion years. And then maybe we might reach consciousness. Who knows? Last two questions. What would you like your legacy to be?
Oh, I'd like to be somebody who, helped get emotion going again, but also that midway through kind of questioned whether we were on the right track. And, my distinction between fear and the defensive survival circuit is one that kind of was a monkey wrench to some of my colleagues in the field. They felt it was a little kind of out of the blue, and but to me it was, I saw things in a certain way, and I felt it was wrong to use animals to develop medications that on the basis of the animal's behavior, when we have decades and decades of research trying to find new medications that were first developed by accident through accidental discoveries in the 60s, and nothing new has really come over all these years. So it causes you to be concerned and think about, well, are we looking for the right thing? And I don't think we're ever going to find a treatment for fear and anxiety by studying an animal's behavior. We have to, fear and anxiety are conscious experiences. That has to be done by understanding the human brain, not an animal brain. Now, it is important because animal research is very important because the behavioral and physiological responses are definitely part of the whole process, but they operate in parallel. They don't determine the state of fear. It's like they control the volume. You walk into a restaurant in the shed back here, and the music is going to be incredibly loud and it's going to be jam-packed, and you're not going to be comfortable. It's going to be unpleasant, but you're sitting at a dinner table. You ask the waiter to turn it down. They turn down the volume, but it's still the same awful heavy metal song. I almost said heavy metal song. It's just not as annoying because the volume has been turned down. I think that's what the behavior and physiology do. You can regulate the volume of the state, but not its content. The content is, what we are having here, an experience between ourselves.
You would like your legacy to be that bringing back to the human brain rather than the animal research?
Well, I don't want to get rid of animal research. I think it's very important, but it's important that we know what we can and can't learn from animal research.
Very important question.
And I want to be known for my music.
And
my heavy metal music.
Who do you think was the greatest musician of all time?
Jimi Hendrix.
Jimi didn't hesitate at all. Why? Why do you think?
Oh, it's just like the most fantastic guitar player ever. Like the genius of guitar. I mean, I love the Beatles too. I like all music, jazz. It's like that. There's a music writer named Greil Marcus, and he wrote books about Bob Dylan and other things. But he had one phrase that really stuck with me. I don't remember where I read it, but he said that the music you listen to as a teenager is the music you will always care about. Other stuff comes and goes. You can get attached to it a little bit, but there's something about that teenage music. And I think what the answer is, teenage years are the time of hormonal upheaval. And hormones not only, make you irritable, they're not just there to do that, but they're there to help you get through the stress and so forth. But, you have sex hormones and stress hormones, and not only do they compensate for stress, but they also contribute to the storage of memories. So all of this hormonal upheaval is like, it imprints the early experiences you've had in adolescence because of their presence. They're at a level that probably never occurs again in life.
That explains why I like all the music I like.
So there's interesting things. So, I was thinking about this not too long ago. I was thought about the fact that, now I listen to a lot of jazz. But what jazz do I like listening to? Miles Davis, all the 60s jazz, like cool jazz, So even stuff that I listen to today, because it seems still fresh to me, was burned in when I was a teenager.
I can relate. Final question. What is the meaning of life, Joseph LeDoux?
Have a good time.
Have a good time. I can get behind that meaning any day. Fantastic. Professor, if people want to connect with you, read some of your books, find out more about you or your research, where can they go? Where can they find you?
Joseph-ledoux.com.
Okay. That's the place to find you. Professor, thank you so much. It was a privilege talking to you.
Been fun.